Hello viewers, welcome back to our channel. Today we are going to see about the blood products, and one is erythrocyte, another one is platelet. First we are going to see about erythrocyte. We are very well known as the erythrocyte is also called as red blood cells, and the nucleus is absent in it. Plasma membrane is very thin, and the cell cytoplasm is covered by lipid bilayer membrane. It does not contain any cell organelles in it, and the shape of the erythrocyte is biconcave disc. Primary component present in it hemoglobin, and the size of the erythrocyte is 7.5 micrometer in diameter. The lack of the nucleus, erythrocytes cannot divide and thus need to be continuously replaced by the new cells synthesized in the red bone marrow. And 120 days is the lifespan of red blood cells, and the development of red blood cells from stem cells occurs in about 7 days. View the process of erythrocytes. The human erythrocytes has a biconcave and discoid shaped and is anucleated. They are approximately 7.5 micrometer and 2.6 micrometer thick and the rim, but only 0.75 micrometer in the center. And the biconcave shape of the RBC cells provide a large surface to volume ratio and facilitates gas exchange. And the average concentration of RBCs in the blood is approximately 3.9 to 5.5 million per microliter in women and 4.1 to 6 million per ml in men. And the erythrocytes are little flexible, which permits them to bend and adapt the small diameter and irregular shape of blood vessels. The plasma lamella or plasma membrane of erythrocytes consists of about 40% lipid, 10% carbohydrates and 50% proteins. A phospholipid bilayer membrane which support the structure of RBC and also cover the cytoplasm of RBC, which is maintained by a network of proteins that make up of cytoskeleton. The cytoskeleton is composed of proteins such as pectins, actin, band 3 and protein 4.1 and ankyrin, which allows for cellular structural integrity as well as malleability. The cytoplasm of erythrocytes lack of all organelles but is densely filled with hemoglobin, which allow the transportation of respiratory gases. And the RBC membrane contains integral and peripheral proteins, and integral proteins distinguish individually in the form of A, B, O and ABO blood types, and they also support the integral structure and bind hemoglobin. And next, peripheral membrane, which help to make the inner side of the membrane and help to make the red blood cells extremely elastic. Now, the function of erythrocytes. RBCs transport the oxygen from the lungs to the peripheral tissue to assist. Next, the cells also collect the generated carbon dioxide from the periphery and return into the lungs from elimination from the body. That's all about the erythrocyte. Now we are going to see about platelets, also called thrombocytes. Discuss in shape 1 to 4 mm in diameter in size. The normal concentration of platelets in the blood is between 1,50,000 and 3 lakh per ml. The shape of the platelets contains compounds exterior coal, cell membrane, microtubules, alpha granules, dense bodies and glycogen particles, dense tubular system and mitochondrion, open canalular system. In thrombocytes, when the peripheral is activated, it anuclear and discoid in shape. The lifespan of thrombocytes is 7 to 10 days and secreted in the spleen. Hypersplenism may lead to the low platelet formation. From the blood stem cells, the myeloid cells are formed. From the myeloid cells, the megakaryoblast cells are formed. From that, the promegakaryocytes is formed. From that, megakaryocytes and lastly form the platelets. Next, The von Willebrand factor and platelet adhesion. In the blood flow, the VWF factors or von Willebrand factors are attached in A1 domains at the both sides. The attached cells are rolled with the platelets and the fibrinogens are formed. After the formation of fibrinogen, the cells get activated and the recruited cells are multiplied. Next, clot retraction. Myosin, actin filaments, and thrombosins in platelets are stimulated to contract during aggregation, further reinforcing the plaque and help releases of granule contents. Now, the summary of platelet activation. Platelets are activated when brought into contact with collagen exposed when the endothelial blood vessels lining is damaged. Activated platelets release a number of different coagulation and platelet activating factors. From the phospholipids, the negatively charged molecules are transported to the platelet surface, provide a catalytic surface for coagulation cascade to occur. Next step, integrins or platelets adhesion receptors. Platelets adhesion to the each other way through adhesion receptors, forming a hematostatic plug with fibrin. And myosin and actin filaments in platelets are stimulated to contract during aggregation, further reinforcing the plug and help releases of granule contents. The GPIIB and IIIA, the most common platelet adhesion receptors for fibrinogen and von Willebrand factors. From the bone marrow, the platelets are formed. Steps: stem cell to megakaryoblast to megakaryocytes and platelets formation. As we know before, the adhesion receptors are formed the hematopoietic plugs. The phases involved in the hematopoiesis: vascular phase, platelet phase, coagulation phase, fibrinolytic phase. What are platelets? In the blood, separated into two forms. One is plasma, another one is serum. From the serum, the cells are formed: RBC, WBC, and platelets. Platelets formation, thrombobiosis, regulation of thrombobiosis by thromboyotin. The major cause of platelet is bleeding disorder. Bleeding can result from the platelet defects. One is deficiency in number, another one is deficiency in function. Another cause of platelet is congenital platelet disorders, disorders of adhesion and disorder of aggregation and disorders of granules. The laboratory testing of platelet function is platelet count, bleeding time, platelet aggregation, platelet function analyzer, flow cytometry, electron microscopy, and granule release products. The last, platelet function. Major function of platelet is aggregation. When the fibrinogen is needed to join platelets to each other, view the platelet fibrinogen receptors. The receptor is fully made up of GPLA, GPVI, collagen, and other components. That's all about the platelet. Thank you so much for your watching. Have a great day.